All right, hi everybody, welcome to your next podcast. And what we're going to focus on today is some evidence that we have of natural selection. So it's kind of a nice compiling or so of several pages in Campbell. If you want more information at any point, you can always read along in chapter 22. So there's kind of four things that I'm going to talk about today that help to give us evidence to support evolution. And there's kind of some themes with those. We're going to look at some fossil record information. We're going to look at some anatomy comparisons that we can make, what molecules tell us with the molecular record, and then a little bit in class in some of the activities that you're going to do will deal with the artificial selection point. So you don't have formal notes on that, just on the first three. But these four things play into evidence that support evolution and a lot of Darwin's ideas. So the first thing we're going to look at here is the fossil record. And Ohio is actually a pretty good place to talk about here because most of the rocks here in Ohio are sedimentary rocks, the limestone that we have. And the picture down at the bottom right-hand side is of Ohio state fossil, the trilobite, which would probably have been one of our pill bugs, except it's about the size of a dinner plate, and that would have been a small one. And that's one of the most common fossils that we have in some of the big limestone sections of rock that's here in Ohio. Ohio is a very tropical place, very warm at one point in our history, and we find a lot of tropical, very large organisms preserved in the fossils there. The problem with using rocks as a record is you can really only look at sedimentary rock for information. Igneous and metamorphic rocks both have in their cycles a point where that rock is actually melted down into a molten form. So whatever could be preserved in those types of rocks is destroyed through that process. Think of a volcano exploding. You don't get fossils spewing out of that. You have molten rock. So it's kind of unique to just look at sedimentary rock for that reason. What's also interesting, and I often teach the little kids to think about, if you get a newspaper every day and after you're done reading the newspaper you stack it on your dining room table. Well, at the end of the week your oldest newspapers are on the bottom of that stack, and the newspaper that you got just that day is going to be at the top of the stack. And that's exactly how we use sedimentary rock layers. Rock layers that are at the bottom are our oldest rocks, and the rock layers that at the top are the younger rocks. And if you have fossils within those layers, it helps you to give a little bit of a time stamp or a date on those fossils. Other things that we can do to learn about fossils is we can look at how much radioactive carbon-14 is in them. We know the half-life of that element, and by measuring how much of that is in those elements, we can kind of get a rough estimate as to how old those are. So just kind of a gallery of fossils. They're very unique through the different parts of the world where you travel. And again, in Ohio, we have a lot of seashell fossils, a lot of leaf fossils in our sedimentary rocks. Thought this was interesting, and any of those Jurassic Park fans out there, amber was a way that we have preserved fossilized insects. This is a dragonfly that we've dated to be about 100 million years old. So pretty interesting stuff. Paleontology can be very interesting and has links to biology in it also. We also had a discovery of a fossil in 2006 that when we look at some of the structures that it has, and that's kind of a nice segue into our next theme, this could be the missing link for the animal that shows us or helps to explain how we started with sea animals and transitioned into land animals. Biointeractive, Howard Hughes Medical Institute has a very nice about 13-minute video about the discovery of this organism. So pretty cool. You probably saw it a little bit in What Darwin Never Knew also. So when we look at these fossil records and when we look at living organisms today, we can compare the anatomy of them. And we're going to first start, there's two types of anatomy structures we're going to compare. The first one's called a homologous structure. Now, what that means is it's a similar structure. So here you're looking at hands, legs, a flipper, and a wing. So if we kind of envision where these are located on the body of these organisms, we kind of have an idea of what it looks like. Also, when we look at how these genes are turned on and turned off during embryology, so this goes on to a little bit of eukaryotic gene control, these all come from very similar sequences in the DNA. So these sequences are turned on and turned off very similarly. Now we get a little bit different look in each one of these. Some are a little longer, some are a little shorter. And what I've highlighted in red are the carpals. So if any of your parents or grandparents suffer from carpal tunnel syndrome, it's where you have a weakness in the tendons and in the carpal bones. And you can see that those yellow structures are in all three of these. What we have to think about here, though, is that these have very different functions. So they've got similar structure, similar development, but very different functions. Now, unfortunately, I know many of you would like to fly, but your arms are not going to let you fly, and I would not advise that you try to fly with your arms. Now, if you're a bat, that's a little different. That structure has a very different function than our arm. So, when we look at structures like this, what it tells us is that we're probably pretty closely related. We probably have a recent common ancestor. And I like the picture of this hairless cat licking its little toes because it kind of helps you visualize maybe that those would be possible fingers. So probably, even though you have similar structure, similar development, you've got different functions, but this tells us that we probably have a fairly common ancestor. And one of the misconceptions that kids have is because I'm always using the picture of these animal arms back here, they think that this is something that just happens in animals. Well, it's not. It also occurs in plants. Spines, leaves, needles, the colored leaves on a poinsettia, the flower is actually that ugly part in the middle. Even tendrils, which helps plants to climb up to reach the sunlight a little bit higher. These are all variations of leaves. So it just kind of brings you into an idea that this not only happens in animals, but also in plants. Okay, so what happens then if you would have similar functions, a very similar external form, but the insides look very different? Well, what that tells us is you're probably not evolutionarily related. And so what this means is that you're just kind of a little bit further apart, a little bit more separated from one another. So a good example to look at here is to keep with this idea of flight. All of these are wings. That's a very similar function, very similar exterior form. You can see what I have there in purple. However, we're looking at three separate animal groups, and the structures of these are very different. An insect wing versus a bat wing versus a bird wing, and I have included some dinosaur wings on here of what we think those look like. The structures are very different. So what this means is we probably don't have a very recent common ancestor. We're not as closely related if we're looking at analogous structures than we would be if we're looking at homologous structures. So another good example to show you with this deals with two different species that swim. We've got fish and we've got dolphins. They're both aquatic. Fish are vertebrates. Dolphins are mammals. So you should recognize those terms in red from your spring break assignment. Very, very similar adaptations to live in the sea, but when we do, and I'll talk about how we know how closely related you are or not, we find that they are far apart on life's trees. So this is kind of always my favorite part of the lecture today, dealing with vestigial organs, and we can make lists and names tons and tons of these, anything from your wisdom teeth to humans having a tailbone, to why when you get goosebumps your hair stands up. You know, those are all things that aren't necessarily very helpful to us anymore. However, there's a reason why we still have these traits. So a vestigial organ, a definition of that is a structure that serves little or no function. And what we think is that this is a remnant of something that used to have some sort of function in our ancestors. So we just have these mutations that have accumulated in our genes, but they haven't reduced our fitness. So they're still allowing us to survive and reproduce. So if you look at snakes and whales, you can actually see remains of a pelvis, maybe even some leg bones. There are fish that live in caves in South America that are blind. They have no eyes that function, but they've got these little eye spots. So the eye's not fully developed, but it's still there. And again, why do we have a tailbone? We don't have tails. And it just hurts when you fall on it really bad. If you look at the vestigial organs, here's the hind legs on the whale, and we even see these in fossilized whale structures also. They look a little different in the fossilized ones. So it's so interesting to study all the vestigial organs that are out there, and if you have any questions about that, please ask me in class. So one last thing to show you about comparing is looking at the embryos and embryo development. And we'll talk a little more about this when we get to the organism part of the course. But if you put a fish embryo, a reptile embryo, a bird embryo, and a human embryo, and get them at all about the same stage in development, they all have very similar structures. We actually have gill pouches, and that's what's highlighted in blue on this slide. Ours will go and develop into our lung tissues, where a fish's develop into gills. And it's interesting to see how the signaling that these cells get determines what they will develop into. Again, if we look at a chick embryo and our human embryo here, we have tail, and a very clear tail. That eventually develops into kind of the bottom of our pelvis and our joints that connect into our legs, where a chicken gets a tail. So it's very different end product, but in the very beginning, as we're starting with those most basic stem cells, we do see some very interesting similarities. So the last thing that I want to show you today is dealing with molecular record. I wanted to kind of tag this in on the end because we are going to be doing a lab later on in the lessons that deals with this. If we look at all life uses either DNA or RNA at some point, and it's used to help code for proteins. It's a universal genetic code. So if we look at some very common genes, cytochrome C is a group of enzymes that's used in respiration, and hemoglobin is used in blood. It's a protein that helps to carry oxygen, so it's used in gas exchange. So very common processes that most life uses. If we compare those common genes, we look at how many different nucleotide substitutions there are, we can kind of begin to develop a record of who's most closely related to who. And if you look at the chart down at the bottom, it's showing you, how many millions of years it's taken to get all these different substitutions that have occurred. So it kind of helps you to line up who's more closely related. And that's exactly what we're going to be working on in class too. We're going to find out how many differences are there in hemoglobin and cytochrome C in different species. So if you compare a human to a human, they're going to be 100% identical. And as you move down to those different ancestors, to those different organisms, it helps to give us an idea of where mutations occurred in those ancestors. And what we'll begin to develop is something that looks like this. We call this a phylogenetic tree, and we start to give a little bit of a numerical value to how many differences we see, and that kind of helps us to categorize who's more closely related to who. Now, it doesn't have to be exact, but it does give us a good indication of some evolutionary relationships. And that's exactly what Darwin was trying to do so very early on. He was trying to build a family tree, and more closely related species would share the same line of descent until they finally just diverge from that common ancestor because there was a different mutation that occurred in their DNA. And that's what we'll be looking at in some of our labs that we'll do later on in the lesson.